talk about biologics or biotherapeutic products. And I think it's important to understand biosimilar concept, to know what we're talking about when mentioning biologics. Now, you all know that for most of last century, and included part of this century, the chemically synthesized medicines, that is, medicines produced by combining chemical compounds, have been or have represented most of the medicines used, prescribed by doctors. Right? And these medicines are, so to speak, simple from their structure. I will not get too technical, but I think it's interesting to go over this in simple terms. So these chemically synthesized medicines are simple in structure and are normally called small molecules. And most of the medicines you know probably are small molecules. Now, in addition to combining chemical compounds to produce a medicine, can we generate a medicine from a living organism? Is that possible? I'm sure you suspect the answer is yes. And we call that medicine biologic, biological medicine, biomedicine, biopharmaceutical, as you wish. But for the sake of this talk and for the sake of your knowledge, biomedicine or biological is a medicine produced in a living organism. That's it, as simple as that. Now, you're probably aware of the fact that insulin, which is a product being used in diabetes for quite a while already, has been for many years obtained from the pancreas, from pigs. So insulin is a biologic. It was obtained from a living organism. I can, for instance, get blood from all of you and isolate a factor called factor VIII and use that factor in hemophilia. Therefore, factor VIII would also be a biological medicine. But can we generate that same human factor VIII in an alternative way? Can I create factor VIII? Not by extracting blood from volunteers, but by other means. And the answer is yes. We can get factor VIII, for instance, from these little guys here. We can obtain the ovarian cells. This is a hamster, a specific race of hamster. And I can obtain the ovarian cells. I can manipulate those cells. And mysteriously, I can generate human factor VIII. So this is a biological medicine because it's been produced by a living organism, by your cell line that we have in culture for years. It's a biological, but it's even more than that. It's a biotech medicine because we've had to manipulate the cells to generate our product. If I extract factor VIII from your blood, I don't have to manipulate you. You produce the factor VIII. If I want ovarian cells from the hamsters to generate factor VIII, I do have to manipulate. So a biotech medicine is a medicine produced by a manipulator or altered, normally genetically altered, living organism with the aim of producing a medicine. All right, so we know what a biologic is, a medicine from the living organism. If we manipulate the living organism and we generate the medicine, that is a biotech medicine. And again, I won't get into technical detail, but just for your knowledge, you might have heard of the DNA or recombinant DNA technology, right? Which is the main technology used to produce biotech drugs. This is, for instance, a cell, right? It could be, as I said earlier, a hamster cell, it could be an insect cell, or a human cell, or whatever, a yeast cell, whatever. I have the cell, and I manipulate the genes of the cell. So I incorporate a fragment here into the genes that generates the protein we want to generate. And as I said, that cells, that's how no matter the origin, will start to produce that protein. So all the biotech drugs that we have now on the market have been generated by this mechanism, by manipulating the gene of a cell and producing what we call a recombinant protein. So most, I would say all biotech drugs are recombinant proteins. And we're talking about black factors like factor VIII, interferon beta, interferon alpha, somatropin, epoetin, interferon beta, as I said, for multiple sclerosis, and all the monoclonal antibodies. Those are all biotech drugs. I'm sure you're familiar with trastuzumab, infliximab, ranibizumab, all the zumabs and non-zumabs, all those antibodies are biotech drugs. Okay, so far so good. It's easy, right? Now, we know what a biotech drug is. Now, as I said, a biotech drug is indeed a protein, and that means that it is a very complex drug. It is very big. This is a typical image, not in scale, but it's a very big drug, and very complex, and very unstable, and with a very complex functional activity. So biotech drugs are far more complex than the small molecules I mentioned earlier. There's a difference in size that sometimes reaches 1,000, 2,000 times, 10,000 times, depending on the biotech drug. So you could ask me, well, so what? It is a drug, right? A biotech drug is a drug, so we should treat them just as other drugs. Well, in fact, that's not totally true. Such complexity makes the biotech drugs that recombinant proteins do have a specific pharmacological behavior. And we'll get to this later. But there's one very important issue regarding biotech drugs that I will explain in a minute, and that has to do with the production process. As I said, we use living organisms to produce them. And there's a very well-known sentence applied to biotechs, which is, the process is the product. What that means is that since biotech drugs are produced in living organisms, they're very sensitive to changes in production. Now, I sometimes explain it in some countries where they really are experiencing the wine production. I compare it with wine. Wine, in fact, is a biological product. You need living organisms to produce wine, right? You need the plants, you need fermentation processes. Now, no matter how hard you try, you know, I come from a country with a lot of wine production, good wine, by the way. Now, if you take the plants from my country and you transpose them to another place, trying to mimic all the conditions, no matter how hard you try, there will probably be differences. So when you deal with living organisms, reproducing something is really hard. Hence, the process is the product. That means that if you change the process of production, even in a very small way, you may get, or you may obtain differences in the end, in the final product. And that's exactly what happens with biotech medicines. Small changes in the production process might change, often very slightly, very subtly, the structure or the features of the final product. That doesn't happen with small molecules. That happens with big, complex biotech molecules. That's the first important message. Look at this, for instance. This is a biotech, the monoclonal antibody, a big antibody produced through the process 1. So we used specific process to generate it, and we changed it slightly, and we attempted to generate the same drug, changing the process. And they look alike, they look very much alike, but how alike are they? And there's here a small change, there's a very subtle structural difference. I made this up, really, but something like this could happen. A very small difference could happen by changing the process. Again, this is something specific or typical of the biotech medicines, not of the small molecules, and that difference is extremely important to understand the message that you will receive at the end. In a small molecule, in a chemically synthesized medicines where living organisms are not used, you can simply generate virtually an identical copy of the active principle. even if you change the manufacturing process. So you can really generate an identical molecule. It's a good advantage. You can, using a different plant, using different methods, produce an identical plant. And as I said, you can't in the biotech world because you may generate small differences by changing the production process. Now, does this matter really? Well, it could, because those small differences may have an impact on safety and efficacy. This is the whole issue. The small manufacturing process of a biotech medicine will probably change subtly the structure, but this may impact on the activity of that. And this is not just literature. That is something that's happened. Let me give you 3 examples, or 2 examples, and then a comment. For instance, myozyme, which is a biotech medicine used for a rare disease, just by changing the production volume. So the company producing that product increased the production volume to meet the demand. And just by doing that, there was an impact on the bioavailability, the pharmacokinetic profile of this product. Just by going through a small, what we call fermenter. So you have these small containers where the cells are producing the medicine, and you want to go to a very big one, and that increasing volume generated differences that impacted on the pharmacokinetics. First example. Second example, efalizumab was produced in a specific plant, biotech plant, and it was changed to a different location, but using the same material. That change of location did induce some changes that generated also pharmacokinetic differences. And this goes on and on. And we also know that for biotech drugs, and don't forget, I haven't even mentioned almost biosimilars so far. I'm talking about biotech drugs in general. For all biotech drugs, there's a batch-to-batch variation that needs to be screened or analyzed because there are chances that changes occur from one batch to another. So this is really very relevant to the extent that when an innovator company, and that happens all the time through the life cycle of a drug, when an innovator biotech company wants to change the manufacturing process, they want to improve the efficiency of the production, they have to meet the regulatory standards, they want to increase production, they want to change the material used to produce, and it's too old for whatever reason. When they want to do that, the regulators will tell them, well, wait a minute, tell me what you want to change, and I'll tell you if there are chances that you obtain something different. And if there are chances that you obtain something different, you might need to do head-to-head studies from the product before the change with the product after the change. And that has happened as well. Amgen is a big biotech company, extremely serious, and they changed the production of a kind of EPO called darbepoetin, that you probably know. They wanted to change some of the manufacturing processes. And the regulator said, well, this is such a big change that we need you to prove that you still obtain the same thing. And they went to clinical trials even to prove that. So they soon did, they discussed with the authorities, and they did some work to show that basically they were obtaining the same way. All right? So first message in biotechnology, in biotech drugs, the process is the product. If you want to change manufacturing process, or if you have to change the manufacturing process, you may obtain subtle but relevant clinical differences. Now let's talk about biosimilars. In this scenario, now we all know this, the process of the product. How does this impact? Now, if you want to think in different situations in which you may want to obtain the same product by changing the process, one of them I've already mentioned it. It is when the innovator company wants to change the process. We've already talked about it briefly. Now there's another situation, and that's when a company wishes to produce that same product once the patent has expired. It is allowed. Market allows that company to do that, no problem. Now, there's a problem. We know that this company that wants to launch an equivalent product will generate probably a very similar one, extremely similar even, but not identical, because it's a biotech product. And you have to ask yourself, what really does it matter? And the answer would be, well, it depends on whether the efficacy and the safety of that product generated by a different company trying to match the reference one is altered. If there's any change in the efficacy of or the safety of that product, why should it matter that there's a slight subtle chemical difference that you can find even in the originator sometimes? So that's the whole basis for the development of the biosimilar guidelines by the European Medicines Agency. And by the way, we do very many things wrong in Europe, but I think this was a good thing. I mean, we pioneered, we established a framework that basically has been followed by highly regulated regions in many ways. So what the EMA says is a biosimilar or a similar biological medicine is the name given initially by the EMA, now it's used worldwide, to a biotech product shown to be similar in quality, safety, and efficacy to a reference innovative product. We could all agree on that. But the most important part of this is how do you demonstrate that? What are the adequate studies? Which is the adequate development of a biosimilar to show such equivalence? And I think this is the difference among different regions in the world. Now, what does the EMA say? And as a matter of fact, what does the WHO as well say? And what does the FDA say? The Canadian agency, the Australian agency, the Japanese agency, all those agencies are very much aligned in the idea of what a biosimilar is and how should it be developed. So what it says, the EMA says, is the following. You have to do head-to-head comparison studies. So you have to take the reference product, you have to take your candidate, your biosimilar candidate, and do comparative studies along the way. And for a generic, which is not a biotech product, and therefore you can't call it biosimilar, which is, as I said, structurally simple, you just have to do a demonstration of physical chemical properties, and finally, when we go to that part, go to bioequivalence. And because the API, the active ingredient, will be exactly the same, if you show it is exactly the same, just a bioequivalence study, a phase 1 study, will be enough to assume that efficacy and safety will be the same. In the biosimilar world, this is not enough. You have to go over demonstration of similar quality, structural, biological activity. Is a very hard and very difficult exercise. But then you have to go through preclinical studies, or you may have to go through preclinical studies. You have to go also through phase 1 studies, sometimes PK/PD. You have to show clinical efficacy and safety in patients, and you have to put in place a pharmacovigilance program. This is much longer than this. This is much more expensive than this. A biosimilar is not a generic. Nothing to do. Let's not call them biogenerics, please, if they're properly developed at least. Now, I won't take much time to show some of those slides, but just for you to incorporate those ideas. When you develop a biosimilar, complex drug, you want to show how similar it is to the corresponding reference product. The most sensitive way to do it is by analytical methods. So there's a wide range of different analytical methods: peptide fingerprinting, biocore, Dacryas, many different methods, molecular mass spectrometry, many different things that will allow you to show how similar both products are structurally. And also, you have to go about what we could call in vitro biological activity methods to show how they behave similarly. So these are the most sensitive ones. So the agency, the European Medicines Agency, puts a lot of effort into analyzing those very first steps of the development. Because if there's any difference, that will be highlighted at that level. So to show similarity between two proteins, two biological biotech products, you have to go through multi-analytical approach, showing by different ways, let's not get into this, that they really are extremely similar. And once you demonstrate that, you can be pretty safe that things will work okay. But still, because this is not a generic, you want to do confirmatory clinical trials. The EMA says, once you've shown such analytical similarity, and this takes years, now go to preclinical studies in mice or in lab animals, but above anything else, show in phase 1 and phase 3 trials that what you have seen analytically, you can confirm it. So you have to go through phase 1 head-to-head trials and phase 3 head-to-head trials. And that's an example of one of the biosimilars approved in Europe. And I just want to show you how the marketing authorization holder did a phase 1 study, that is obviously all this before authorization, with 44 individuals, a second phase 1 study with different endpoints, I will go over it, with 48 individuals, and a phase 3 study with 250 patients. And that was a relatively simple product in the biotech world. I mean, this was not a very complex medicine. But still, look at the whole amount of clinical studies needed to be done, and that's what makes it extremely expensive to develop one single drug. And they showed, by different means, that there was overlapping of the clinical outcomes between the biosimilar candidate and the reference product. So I won't get into detail, but what those graphs show, these are selected graphs among hundreds of them, show how the behavior in the patients, the efficacy, safety, and the pharmacokinetic level was extremely similar.